I'm going to start recording, but I have to write with my finger until the pencil's charged, so it's going to be messy. Yes, please close the door and turn the lights off. So when you're looking at a project like this, you really should be reading, because the reading actually helps a lot. It says protein synthesis is a process used by the body to make proteins. I think that should be obvious. I don't think that, but I've had people ask me, what is protein synthesis? What does it do? I've had people ask me that. I'm like, really? It's in the name. The first step of protein synthesis is called transcription. So that's the one word you should all know, right? Transcription. And then during transcription, and what happens during transcription? mRNA copies the DNA. It's changed a little bit. The DNA is unzipped, and mRNA copies are made from a strand of DNA. That's transcription. Once it does this, mRNA is going to leave the cell. Now, remember, there's a whole process that we discussed, and you should know the steps. This is an overview. We'll go over it later, but just so you know that there are many steps you should know. mRNA will then attach itself to a ribosome outside. That's a different step. What's that step when it's happening outside? When mRNA attaches to a ribosome, what's happening? It's not transcription, it's translation, correct. mRNA strand is read and make copies. There's three bases at a time that we know as codons, right? So this is an important point here that these things called codons, and tRNA is the stuff that fetches the amino acids or brings the amino acid over, okay? It brings amino acids to ribosome. One mRNA strand, there are start and stop codons, and they should all be in that chart that we reviewed, that you will always get on a test. You don't have to memorize it. You can look it up. It's called the Universal Amino Translation Chart. It's actually two versions of it. One's weird and circular. See if I have a copy here somewhere. Here's a copy of the chart. This is the simpler chart. I like this chart, but there's a round one that you'll see that we'll need to use, but you have this chart. It's called the Universal Code. Remember, amino acids are the building blocks of proteins. We went over that, peptide bonds, amino acids. We went over the basic structure. You do have to know the basic structure of amino acids, of carbohydrates, of lipids, and of nucleic acids. We went over that a lot. You should know what those are. So in the nucleus here, inside the nucleus, this is, of course, only in eukaryotes. In prokaryotes, they do transcription translation, but it all happens in the cytoplasm because there is no nucleus. In eukaryotes, you have DNA, it unzips, and it starts reading, and you make an mRNA from one of the two sides, and the DNA is read. mRNA is made by DNA polymerase II. And that RNA that's made there then will be processed after transcription. It'll put a 3' cap, it'll put a poly A tail, and then that will leave the nucleus. Introns are taken out, exons are spliced together, all that stuff that we talked about. That mRNA will leave the nucleus and go into the cytoplasm, where we'll find a ribosome. And remember, this is going to be a key point. I can almost guarantee it's going to be on the test. If the mRNA has a specific sequence of RNA, then it will attach to the endoplasmic reticulum. The ribosome and mRNA will attach to the endoplasmic reticulum, and then that protein will leave the cell. It will be excreted, or it will go into an organelle, inside an organelle. If it doesn't, then it's made in the cytoplasm, because that protein is to be used in the cytoplasm. So there's a decision-making point. There's a fork in the road at that point when you're talking about mRNA, it has a choice. Is it going to be used in the cytoplasm? If it's going to be used in the cytoplasm by the cell, then it'll be made in the cytoplasm. That should make sense. If it's made in the rough endoplasmic reticulum, then it's leaving the cell or going into a membrane. Or inside, sorry about the messy handwriting, I can't do anything without my pencil neatly. Or in a membrane or into an organelle. So there's a sequence, and we don't need to go into it, but basically they're hydrophobic amino acids are coded for early on in the protein. Then it'll generally be attached to rough endoplasmic reticulum, where then it will continue to be made, and it'll go into one of these three places, A, B, or C. So RER is part of that endomembrane system that we discussed, that you had quizzes on, you should have notes on. All right, what's the first? Now let's go ahead and answer the questions. Oh, before we do, this is translation. So you have the tRNA. I think it looks like a T to me. So the tRNA it's T though because it's transfer RNA. Transfer RNA. And the transfer RNA brings the amino acid, it transfers the amino acid from the cytoplasm to the ribosome. The mRNA has the messenger RNA, mRNA, and the ribosome is made of RNA as well, and big and small subunits, and that acts as the enzyme. So it's going to read and help the amino acids to form peptide bonds. Is that clear? That's the process. This is all happening in the cytoplasm or on the rough ER. Now, most of your questions at a regular high school level, this is an honors class, correct? But at a regular high school level, there would be enough to know it's cytoplasm. So when you take the end of course exam, it's enough to know that translation happens in the cytoplasm. But in honors, you should know, I think, that the ones that are going to be secreted, like insulin, mucus, like when you get a snotty nose, all those proteins are made in the rough endoplasmic reticulum, and that's how your body knows these proteins are to be put out of the cell. And so you get a finished protein that will wrap up, right? Have some kind of secondary, tertiary, quaternary structure that'll have a specific shape. Remember, biology shapes are so important. That's why art is so useful in biology. The first step to protein synthesis then is what? Yeah. Transcription. Easy peasy. Lemon squeezy. I'm going to see if my pencil works now because I can't stand using my finger. All right, so transcription. All right, what is the second step of protein synthesis? Yeah? Translation. I want to remind you that on your test and your final, transcription is going to be broken down into a series of steps, right, that will ultimately lead to translation, that will then go off, and that will make some other steps that ultimately will make a protein. Also, one other reminder, because I could see this coming up on the test as well, this is just a reminder, not all genes code for proteins. Not all genes code for proteins. Let's be clear on that. Some genes make ribosomal RNA, some genes make tRNA, some genes make other forms of RNA that do other things. But all genes make RNA. Some of those genes in fact, most of the genes, most of the gene products, most of the gene expression, most of the work is done by RNA. So you go by mass. If you go by mass, most of the RNA is not mRNA. Most of the RNA is not mRNA. It's tRNA and rRNA, which is ribosomal RNA, and tRNA, the transfer RNA, and other RNAs. But the mRNA makes a lot of protein. Every one mRNA can make many, many proteins. So most of the work in the cell is done by proteins. Does that make sense? All right, so I just want to be clear on that. When we go by mass, most of the RNA is tRNA and rRNA. But when you go by work, it's the proteins that do the work, and the mRNA are essential to getting the proteins made. Yeah?
On the test, would we have to identify what like look at a picture and identify what
Yeah, I wouldn't say that's not impossible. And I think tRNA, looking at tRNA, I think that's easy. I think tRNA looks like a T. I think that's easy. It's tRNA. mRNA is going to be easy too because it has a code, AUG to begin with. Not difficult. rRNA is going to look like a ribosome, right? Look at it. I mean, you could tell the difference, right? I could see, and I'm not saying this is what's going to be on the test. I haven't made the test yet. But I would say if you got a picture like this, and I had labels A, B, and C, I think it would be fair for you to be able to say tRNA, rRNA, and mRNA. There's 3 kinds of RNA right here in this one picture. I think that's fair. So yeah, you might be asked in those shapes. All right. Where does the first step of protein synthesis occur? Anybody? In the nucleus, right? Excellent. All right. Where does the second step occur? In the cytoplasm is fine. Now, I want to say, remember, RER or cyto, right? Don't forget that. That's kind of a little bit of an addition than a regular biology class would have to deal with. Nitrogen bases are read, how many? 3 at a time in translation. Now, this is key. They don't tell you this, right? It's 1 at a time in transcription, but in translation, it's 3 at a time in these things called codons. They're read 1 at a time in transcription and in DNA replication, which we haven't covered yet. The bases on the mRNA stand for what? Codons, right? The bases on tRNA are called anticodons. Remember, they're triplets. Right, triplets. What is the start codon?
AUG.
It's always AUG.
But I got a question.
Yeah.
I think I already asked you this, but I forgot.
All right, go ahead. And this time, write it down. So you don't forget, because you will.
Start AUG? I think I asked you that. Right?
Yeah, it's a good question. Always starts AUG. So if you have this long mRNA that has a bunch of different nucleotides, it's at the first A, U, and G. The first AUG, the first time you see first time it sees A, then U, then G, that's where the ribosome is going to start doing its job. This other stuff is not going to be part of the message. Now, I say that to you, and I've seen plenty of questions where they don't even bother dealing with that. They'll give you an RNA sequence. In fact, I think we're going to do some right now. They're going to give you an RNA sequence, and they just assume you're going to start wherever they tell you because it's part of the problem. So when you do a multiple choice problem or fill-in, sometimes they won't start at AUG, all right? So you got to know that you have to put your knowledge to the test. And when you're given a question, you have to contextualize your knowledge. So you have to look at the question. You have to say, okay, in this case, they're not asking me about the start codon. They just want to know if I can take DNA and make RNA, and they want me to know if I can do it right, so let me do that right. But sometimes they might ask you AUG, so be careful. You have to be careful. I wish they would be consistent, but they're not. Yeah. Yeah, you'll always have it. I've never, even in college, in med school, I have not seen anyone say, here, memorize this chart. No, nobody, not even biochemists memorize. I mean, they'll have pieces and parts memorized, but they're not going to memorize all those combinations. Yeah.
Okay, just make sense. So if it was like GCG before that, I would skip that and start.
Yeah, if it was GCG before that, this would not count. You would not this would not be part of your. Now that depends, like I said a minute ago, that depends on the question. All right, so read the question carefully. I can't emphasize that enough. They love to throw, and by they I mean the state of Ohio, I mean the AP College Board, the SAT, ACT people. They love to throw these kind of like curveballs at you. All right, yeah. No, but we'll get on to that. Let's go on. If you did you say what's the start codon? Did you put met here? Yeah. Oh, that's fine. It would be wrong, but make the correction. Okay. AUG, the codon is not the amino acid. A codon means the sequence, the triplet sequence. Know that. That codon does, exactly as you say, mean, AUG does mean methionine. That's what's going to be on the tRNA that matches AUG. By the way, codons are on the mRNA. I know it says it here, but be sure that you understand. Codons are on the mRNA.
Wait a minute, so we don't put MET?
Not here, not to answer this question. This question is, what is a start codon? But yeah, you would put MET if you were translating it, yes. What are the stop codons? Well, there's 3 of them. What does anybody know what they are by offhand? Yeah, UAA, UAG, UAG, and what's the other one? UGA. Now, would you have to memorize that? No. No one's going to ask you to memorize even those 3. They're going to give you the chart, and the chart tells you UAA, UAG, and UGG, or UGA. All those are the 3, and they're in the chart. There's always going to be 3 stop codons and 1 start. That's it. It really is easy, especially since you're always going to have a chart. A bunch of amino acids attached together is called a what? Well, yes and no. It's called a polypeptide. If you put proteins, it would not be wrong. A protein can be one single polypeptide, but a protein can be multiple polypeptides. Is that clear? But it's certainly a polypeptide. So it's not, it would not necessarily be wrong. I wouldn't mark. All right, so when we're looking at this, remembering, let's write down what I said because some of you are just staring into space. Let's make sure we understand that alternative splicing can get one gene to produce more than one kind of protein. So that's important. And remember what else splicing is. When you have DNA and you have this ATGCAT, you're going to have these sections of DNA. They're going to be cut out. They're cut out, and then the other 2 sections are put together. These are called introns. And honestly, I wish they would not have called them introns because to me, in means they stay. I understand why. And these are exons. They're called exons because they're expressed. They're sections of DNA that are expressed. They actually become part of the RNA, where the introns don't. But we can cut them up this way and make one protein. We can cut them up a different way and make another protein from the same gene, from the same DNA sequence. Other organisms don't do that. So alternative splicing gives us an advantage. But we're not looking at splicing here. We're not going to look at splicing. These kinds of problems, they don't make it that complicated. We're going to make it simple. And on your test, when I ask you to do this on the test, it's going to be this simple. It's going to be as simple as I'm doing right here. So I'm going to go ahead and do it. And I'm going to say DNA to RNA. CCT gets transcribed to what? And translated to what? What would it be on the mRNA? Transcribed. It's transcribed. It would be GA. And what's the next one? GAA. And what's the next one? AUG. And what's the next one? I don't know what's going on over there, but we can stop anytime now. That would be great. 1, 2, 3, 4. We can stop anytime now. UGU. And then what's the next one? GCC. And what's the next one? UCC. And the next one? CAG. Next one? Oh, CAU. Is it CAU or CAG? What is it? CAU. I'm trusting you. I can't see. I'm not following. So, which is going to translate into what? The next one? Transcribe into what? G, C, G. And the next one? AU what? A. Next one? AGA. Next one? UAC. And the next one? Are there any others after UAA? What are they? Is it a stopping one? Is it not a stopping one? That's my question. So where would I go? I would go here.
Yes, it's stopping.
But it would still turn into RNA. It would the UAA still gets turned into RNA, but I'm just telling you it's a stop. So it would never get translated into an amino acid, but it still goes on. After UAA, what is it? UGU. And then what? GCC. Then what? UUC. UUC. And then what? GCU. What is it? I'm sorry. GCU, and then what? UGG. UA what? And then what? UAG. And that's it? So that would be the mRNA. I had to twist it around so much. But now we have to take that and turn it into an amino acid chain, because that's RNA. That's where we went from DNA. We went from DNA to RNA. That's transcription. But when we want to go from RNA to protein, that's translation. So now we're going to take that mRNA and turn it into an amino acid. If you're not following along, you're having side conversations. Wow, I don't even know where to begin, except to say you're going to end up in trouble. I don't know what, I can't. All right, so when we look at this, GGA gets turned into what amino acid? Oh my goodness. It's a spam call. I'm not going to deal with it. All right, GGA gets translated into what? Now we're doing translation. We just did transcription. We're doing translation. What is going on? Mr. Mendoza's room, may I help you? Yeah, you got the wrong number. Please take us out, whatever it is, off the list. Thank you. Bye-bye. I hang up on purpose, lady. All right. GGA gets translated into what? What is it? GLY. Do you agree? GGA, so G, first is G, then G, then A, gets GLY, glycine. You're wrong. You're wrong. It is GLY. GGA is GLY. You're right. But is not GLY at the same time. Why? It doesn't start with GLY. What does it start? AUG. So it starts here. It does not start here. It starts here. I'm going to warn you again. Be very careful. Some questions have you start in the middle of translation. Others ask you to start at the start of translation. They gave you an example, didn't they? AUG is the start. They gave you that example. Follow the example. So in this case, AUG is the start. So here is where it starts. Now from there, UGU is what? What's UGU? CYS. Let's double check. UGU. If you don't know what's going on, pay attention what's on the screen. UGU is CYS. Is correct. It really is the easiest thing you're going to be able to do on the test. I mean, except maybe for osmosis and diffusions, it's also easy if you know what you're doing. Those are really easy points. I can't see why anyone wouldn't get almost 100% on this, if not 100%. What's GCC? What is it? SCR. And UCC?
That's SCR.
Oh, what's GCC? ALA. And UCC is SER. And what's CAU? What's CAU? HIS. I'm just going to, I have to continue. Histidine. GCG is what? Still nothing? Alanine. Do you guys not see it? GCG is alanine, right? ALA. You can just say ALA. AUA is what? Isoleucine, ILE. AGA is what? AGA is arginine, ARG, right? And UAC is what? Tyrosine, TYR. And UAA is what? What's UAA? Stop. That's right. So if you have a hard time doing this, you need to watch some. At this point, you can't come and see me today after school because we have the neurosurgeons coming in for neurosurgery opportunity. By the way, if you guys want to come in and participate in that, you're welcome. But that's this afternoon. So I won't be available for you this afternoon. I will be here tomorrow morning, and we'll be here tomorrow afternoon. So if you need help, come and see me. But you should be able to do that. All right, if I have time, I'll come back to this, and we'll continue recording. We'll finish this. So I'm going to go ahead and, if you don't mind, I'd like to answer these questions first, and if we have time, go back and get the other ones done. Because those other ones are going to be pretty much the same thing, right? Circle the correct choice within the parentheses for 1 through 18. DNA or RNA can leave the nucleus. Which one can leave the nucleus? RNA is correct, so that's the correct one's RNA. mRNA is made during what? Transcription is correct. mRNA is made in the nucleus. So yeah, it is used in the cytoplasm. So whoever said cytoplasm, it's used in the cytoplasm. But it's made in the nucleus. Very tricky question. DNA is located in the nucleus is correct, and only in the nucleus. In eukaryotes. Remember, I keep saying this, but in prokaryotes, The DNA, everything's in the cytoplasm. There's only one space in the cell. There's not multiple compartments. Translation, transcription. Transcription converts DNA into RNA. mRNA or tRNA? Used to carry. Carry is tRNA. Is used to carry the genetic code. Oh, it's mRNA. Sorry. Carry the genetic code. I saw carry. I thought amino acid. See. That's how you get. See why it's tricky. Your brain fills in the blanks. I'm gonna tell you, when you're in a rush on a test, that's why people make mistakes. The people that study, that actually do prepare, you tend to make one or two small mistakes. But you can do it as long as you take your time, go back and check, so you catch these silly mistakes. tRNA, RNA makes up a ribosome. tRNA or rRNA? rRNA makes up the ribosome. Guys, that should be, I think that, look, R in ribosome, R in RNA. In fact, that's what the R means, ribosomal RNA. That's what the R stands for. You should write it down so you don't forget it. rRNA is ribosomal RNA. tRNA is transfer RNA, but it looks like a T. DNA or RNA uses uracil instead of thymine. RNA. Blank. RNA or amino acids make up a protein. Amino acids. Transcription takes place in the nucleus. tRNA is used in translation. Now this is tricky because all of these begin with T. Remember, translation is where you put the amino acids together. So that's what tRNA does, is carry amino acids. tRNA uses anticodons or codons to match the mRNA. Anticodons. The codons are on the mRNA. Use the mRNA to read. Ribosome, rRNA uses mRNA to read. Proteins are made in the ribosome. tRNA or mRNA attaches to the amino acids into a chain. Well, that's kind of weird. I don't like the question, but tRNA is what I would choose here as well. It's just I would say it's the ribosome that's doing that. That's the enzyme. tRNA is just holding it, but okay, that's fine. tRNA is found in the cytoplasm. Yeah, it's made in the nucleus, interestingly, and then it goes into the cytoplasm and pick up an amino acid. Translation, transcription converts mRNA to a protein. mRNA to a protein. Oh, whoa, what happened? Oh, I hope the battery's not dying. Someone turn on the... Oh, well, no, we'll wait and see if it actually turns back on. Like, why would it blink off? I dusted that out when I first started the year, so I can't believe it's dust. Please start. Please work. Please. No? Not gonna. All right, let's hope. You know, this month of December is going to go down in the history of my life as horrible. I mean, let's review. Death in my family. Shock death. Like I have to leave school and go deal with the death. Absences. Stuff that happened to my brother happened this year as well. A lot of it.
Wait, was that this month?
Some of it happened this month. My brother having going to ER, ICU. That was right before my mother-in-law. And then I come back, the first day back from dealing with my mother-in-law's funeral. What happens? Somebody threatens to shoot us. I mean, my God. Could the universe please back away? And then the computer doesn't work. The printer's not printing. And now, finally, we get the lamp to work again. Thank you, Jesus. Thank you for give me a break. All right, so transcription and translation converts mRNA to a protein, mRNA to a protein. Translation. Transcription is DNA to RNA. Make sure you get that straight. I know it can be confusing. That's why we do this. It's translation. Translation is making the protein. Translation takes place in the cytoplasm. All right. Okay, so now we have DNA. They tell you this is DNA here. They tell you this is mRNA here. And they tell you that this is tRNA here. So this is the anticodon is going to be here. The codon is going to be here. And then the amino acid sequence is down here. All right, so at each step, and we're going to go ahead and pull this down. Let me get this. Where's the chart? Oh, I guess I have another page. I'm going to have one of these charts on every page. All right, so again, this is just what we did on the other page. Not a big deal, right? So we would have to start with the first one being A gets turned into U, C gets turned to G, A gets turned to U, A gets turned to U, C into G, or G into C, A into U, C into G, G into C, G into C, T into A into G, to U, G into C, C into G, thank you, T into A, G into C, and A into U. All right, so there it is. That's simple. Now the tRNA, the anticodon is the opposite. So U goes to A, G to C. So actually, the anticodon is going to be the same as what? The first, right? Now, it's not going to be the same code, though. And the reason it's not going to be the same code is because you have... let me finish this because I hate doing two things at once. Two things make it different. Two things make this different, and the two things that make it different is obviously that where there's a T here, there can only be a U here, because this is all RNA. The other thing that makes it different, and they don't show it here. They're not showing it here, right? So let's be clear. They're not showing it here. What would happen when you're going from DNA to mRNA is you would have parts of the DNA code, parts of this code would be taken out, and the rest would be spliced together. So this code would actually be different from this code. Is that clear? That's really important, especially for creatures like us that are very complicated. This becomes very important. That explains how 10,000 genes can form many, many more different proteins than they could if they just did a one-to-one transcription. By allowing us to transcribe the gene in different ways, we're able to have one gene make 3 or 4 different proteins that do under different conditions. Does that make sense? So alternative splicing, putting these together, changing this code here at this point, makes us able to do a lot of the things that we're able to do. In fact, some people think that our intelligence as human beings is partly due to how we change the code during this transcription. So our DNA is very similar to chimpanzees. We're 98% similar. We have the 98% similar genes. But what we think makes us so much different, what makes us really different from chimpanzees, is that how we change the code after we make the mRNA. Before we make the mRNA, so we make different proteins. And those proteins then make us that much different. Does that make sense? So we share the same 98% of the same DNA, but we process it differently. That's one of the big differences. All right. So somebody tell me the amino acids. And as other people check, make sure you agree. THR, ARG, ARG again.
Wait, do we leave that? Do we keep the C there or do we leave it?
Oh, there should be a C here, yeah. I hope you're reading this. When you're translating this, I hope you're reading the mRNA, not the anti. Don't read the anticodon. There are charts that are made for the anticodon, but they're for specific biochemical uses. For most of the charts you'll ever read will be an mRNA chart.
So we're going from the
mRNA. So UGU is cysteine. So this is wrong. I thought that might happen.
So we always go from the
That's right, and that's why one of the reasons I want to make sure I did this. So you always go from the mRNA. So UGU is CYS, and then UCU is SER, and GCC
it's
is
Allah.
alanine, and AUG is methionine. Now, if you get a start in the middle of a sequence, it's not a start because it already started, right? It's methionine. And so ACU is threonine, THR. So you see, you can get radically. Do you see why I wanted to make sure you had a problem like this one? Because I could tell, I've seen this actually on an end of course exam, and I've seen kids actually use this anticodon instead of the codon, and they got it wrong on their practice ones. All right? I'm not going to be there when you do your end of course exam because I tend not to I try not to be in the room when you're doing I don't want anybody to say we're cheating. So, but during practice questions, I've seen kids make these mistakes. All right, so don't do it. All right, so if you want me to correct it, I'll go ahead and correct it now. So what this should be is U goes to A, C goes to G, G goes to C. There should be a circle here. There isn't. That was a mistake. C goes to G, C goes to G, A goes to U, U goes to A, G goes to C, A goes to U, C goes to G, and U to A. If you want to be 100% accurate, that's the way it would look. But again, the important part is you get the mRNA because the codons are read from the mRNA. So the codons are going to be UGU, UCU, GCC, AUG, and ACU. This is what you're going to read. Let me use the highlighter. This is what you're going to read in triplet fashion off of this chart to get these amino acids. Just be clear on that. Last set of questions here before we go back if we have time. Draw a DNA nucleotide and an RNA nucleotide. Label each of the three major parts. This should be easy, but it's important that you're able to do it. So obviously you have the ribose sugar, correct? And the oxygen's there. This sticks up here. This sticks up here. So we have a 6-carbon sugar. We have hydroxides. You don't have to worry about all the OHs and Hs. Let's not worry about that. But if we're doing a DNA, deoxyribose, that means there's no oxygen here. You should put an H, make sure there's no oxygen. And when the big difference between DNA and RNA, remembering, is that you still have the same ribose sugar. 1, 2, 3, 4, 3, this is 3, 4, 5, and there's 6, right? So there, 1, all right. And on the second prime, let's erase these. Just not so we don't get anyone confused. We're looking at this. This has an OH on it, right? There's a hydroxide on that. So that's the difference, one of the big differences. This up here is connected to, this carbon up here is connected to a what? A phosphates, correct? And then over here, this is connected to some kind of nitrogenous base, either single ring or double ring structure. This over here is connected to nitrogenous base, and I'm not going to draw, I'm never going to ask you to draw all the details of the nitrogenous base, but it's also connected to a phosphate. And the difference is that this nitrogenous base is either A or G or C or T. This is either going to be A, G, C, or T. Where the other, bless you, the other, in RNA, it's either going to be A, G, C, or U. It's either, again, it's either going to be A, G, C, or U.
You said NB stands for what?
Nitrogenous base. NB. What is the point of DNA replication? Make copies of DNA. Now, we really haven't gone over DNA replication, so I'm not even sure if these are appropriate questions. When and where does replication occur? In the nucleus. Has to happen in the nucleus. Before mitosis / meiosis, during S phase. You don't know any of this, so you don't have to know it. I'm just giving you the answer. All right, you don't have to know it for the test. We haven't covered it yet. Are we clear?
Yeah.
What is the point of transcription? Make RNA from DNA. Where does it occur? The nucleus. What are the three nucleotides together called? What are three nucleotides together called on mRNA? A codon is correct. And where is the codon? On mRNA. And that was a codon chart we were reading. So that's why you're always reading an mRNA. The mRNA codons can be used in the chart to find what?
The sequence of amino
Amino acids. You can put sequence, or you can, or amino acids, right, that are going to be replacing it. But you're right, it would be the sequence ultimately. What molecule contains the anticodon? tRNA. And during what process is it used? Protein synthesis. What specific? To make proteins, what's that process called when you're making the actual protein? Translation. Mm-hmm. Translation takes place in a ribosome. Again, I said this twice before, but I'll say it again. That ribosome can be in the cytoplasm, and usually that's the answer you're going to give, or in the RER, right? And blank make up ribosomes.
RNA protein?
No, it's rRNA, and proteins is fine. What is the point of translation? Make a what? A protein. You can also put polypeptide. The best answer is a polypeptide.
Can I resubmit this?
Yes. Transcription and translation together is the process of protein synthesis. All right. Again, that's it. Are we good? All right, so we don't have time to finish the other questions, right? All right, 5 minutes, so we can finish some of them, right?
We answered everything.
So I don't want to rewind this because I don't want to go back and mess up the recording. So I'm going to stop here, but I'll go and fill in the other answers.